this will be interesting for those who are interested in elevating the brain function, possibly improving IQ. But if you're a meditator, I think you'll see why possibly using photobiomodulation the way we are trying to do it can elevate your experience. And elite athletes, or you're just active and stuff like that. So, but my title is talking about parameter settings. They're all connected. Now, parameter settings is going to be quite important. In the past, it hasn't been dealt with much, but I'm going to show you quite a number of brain groundbreaking stuff. Photobiomodulation, more popularly known as red light therapy, has been around now. It's been up in the headlines and even in scientific journals, popular journals, social media. So it's doing some good. Generally, I'll say photobiomodulation seems to work in aesthetics, in, say, hair growth and a bunch of other things. If it doesn't, then it wouldn't be around that long and getting that kind of attention. We are here. Now, what we want to do here is that I want to try to get to be the best in trying to optimize your experience and outcomes. So it's got to be personalized in some way. So we have to do a lot of research to try to determine what's working. Now, the parameters for photobiomodulation has been done over the years. It was first discovered and published in the Russian language in Hungary in around 1967. So quite a lot of work has been done because it's such a mysterious thing, low-level laser activating good outcomes and so on. It started with. Then it got quite a lot into cell culture studies, animals, then progressed to lasers. Now we have large arrays of LEDs for brain stimulation using photobiomodulation. But you can see, my opinion, the most precision came out of cell culture because then we can nail down what is the dose, right? When it comes to animal study, it's less precise, lasers for the human beings, and then with large array of light to the head, it's actually even less precise. So generally safe, efficacious, but imprecise. How do we get more precision? This is a window of where the efficacious parameters of photobiomodulation would be. It's a complicated kind of scientific language. It's biphasic dose response, or in the Anschutz rule, saying that there is a window where outside of the window in either side, whether it's too low in terms of delivery of the photons or too much, will have either no effect or negative effect. The dose is really a function of delivery of the power to your target surface. It's called irradiance, and amount of time. Then you get a dose, or when it's calculated on the surface of your skin, is fluence. So they're kind of like nuanced differences, but generally this is the idea. But actually there's a much more dimension to this, because these other parameters, like whether it's a laser or LED, the wavelengths, whether it's red, near infrared, or whatever, location or position of where you're directing the light, power density, and I say power density has a lot to do with contact, and pulse frequency. This is something that we are really into right now. So back to cell culture. Now, when they did the cell culture, and I say it was precise, actually the dose was around 3 joules. 3 joules is just about enough energy to heat 1 gram of water to 1 degree. So basically you don't feel anything. Now, how do you translate that to an animal, a human being? You got to penetrate like the scalp, right, and get into the brain and all these various parameters. So it's complicated. Let's go back to these parameters now. And I'm saying dose is what you're looking for is a function of the power density and over the time. So we get a dose. So let's try to nail down the time. This is an analogy I've used before. You put a chicken hug it, some people might like roasted, what inside is light, but if you roast it at the right temperature over 20 minutes, yeah, you get evenly distributed cooked chicken. But I'm really interested in what it means in terms of gene replication, the expression of genes. And I'm probably the first one to show this. And going back, it's not obviously not my work. 2 years after the DNA, the double helix was discovered, Masterclass and Stahl in Caltech actually, they did this experiment on bacteria and see how long it takes for the gene to be replicated. So, and what they used was a sample of E. coli bacteria. It took 20 minutes. But human beings and all, it takes longer. So it's kind of a benchmark. But I know 20 minutes is used in medicine, in alternative medicine, quite a lot. So that's kind of a magical number. So we stay with 20 minutes, but there is a genetic explanation for it. So is it laser, LED? And I'd say, personally, we use LEDs. Everybody out there, except for clinical use, having to need FDA clearances, use LEDs. In photobiomodulation, I see this as quite a bit of a benchmark study. We say that really, it's not really, the outcome is not really dependent on whether it's laser or coherence, which is laser is coherent and LED is not, it's divergent. And it's really to do with the wavelength, power. Laser is coherent light. Now light travels in waveforms. So it's coherent. They're kind of going in steps. Now LED is spread out. So when it comes out of a power source, it just goes, spread out. So when you are at a distance away from the source, you can see that the power density is reduced. You can point laser like I am going to do. So it's quite a distance, right? You still have a small diameter. This is a laser. But I point an LED light, you're not going to see it on the wall. So that is the difference. The distance matters. How do you solve it? Now, if you put your LED source right on the surface, you are very much reducing the difference between the laser and an LED. So contact is really important. You can calculate it. It falls into the inverse square law for radiation. You literally square, Of course, it comes in the ratio, but it just gives the message that distance has an exponential effect on the power you're trying to deliver on the surface. And this is just looking at illustrating it. When you have space in between, that means that's important. Now, you can bring sunlight into the equation too. Actually, if your LED hasn't got enough power, you get, I'm always an advocate for people to go out in the sun. The sun's got full spectrum, it's natural. However, if you stay too long, you're at risk of exposure to ultraviolet and you have other issues with the skin, right? But the sun does have merit. And I would say if you want to deliver like 100 milliwatts, and I'll show why I'm interested in 100 milliwatts per square centimeter density, you want to be like, sometimes it's difficult to be in contact, but if you're away, say 1 centimeter, for 100 milliwatts, then you probably want to start at like 300 milliwatts. So it's powerful. You cannot get these LEDs off the shelf. It's got to be engineered in some way. But if you really want to get this efficacy, you do that and you go further out there. The average irradiation from the sun is really about 100 milliwatts, generally average. We measured in a spring day when spring came about in Toronto, it's about 50, we measured 52 milliwatts, but it could be brighter somewhere else. So that is a feel of what it's like. It's always somewhat subjective. So distance plays a significant role. Now we have covered these other parameters. Now pulse frequency is where things get really interesting because pulse frequency delivered through photobiomodulation, that seems to affect the waveforms, both the oscillation and the network. And we are learning this from cell culture studies of living cells like tubules, microtubules. We are doing this with the University of Alberta, with a bunch of molecular biologists, physicists, quantum physicists. Now what we found is interesting. There seems to be an increase in resilience in the cell, but this on the living cells. But I want to look at the tubulin conglomerates together or polymerize together, aggregate together to form microtubules. Many of you are probably familiar with microtubules. They form the structures of the cells, the axons, the white matter, and so on. Now what we found was on the control versus the exposed tubulin, actually the density was lower. So it seems to be scattering in some way, causing a transition in the tubulins that were supposed to polymerize to microtubules. Then we are writing out a paper. When we pulse at 40 hertz, gamma, it looks quite different. It looks like almost as the control and exposed microtubules looks to have about the same density. And when we increase the frequency, like 1,000 hertz, it starts to go the other way. So pulse frequency does matter when you expose like microtubules and tubulins to pulsing light. At least what we used was 810 nanometers. There's something to be learned here. Then we did Raman spectroscopy, which is an instrument to look at the structure of the protein and structure of the cells and so on. Now we found that 10 hertz is actually causing the shift. It seems to be causing the microtubules substructure to transition. Well, basically it's alpha helices to beta sheets. But the bottom line is this is a message to say that when you pulse at 10 hertz, you are actually destabilizing the microtubules. Is that good? Is that bad? But it's causing this. I know 10 hertz, right? When you look at subjects who was reporting they sleep better, they're more relaxed, and so on. So the picture can be quite complicated, but that's also to do with the network on a network basis, which I'll touch on later. So in summary, 10 hertz is causing transitioning in microtubule structures. 40 hertz is starting to consolidate. If you do 10 hertz, you are kind of priming, in a way, priming your brain. If you add another intervention, it could be neurofeedback. Maybe then you are actually having a brain structure that's more welcoming. If we do gamma, it seems to consolidate the structure and beyond. So we also do EEG. We published this in 2019, and we were showing for the first time that when you deliver gamma to the brain, it does an unusual thing. Not the whole brain goes up to higher frequencies. We actually see alpha, beta, gamma having more power and then down-regulating delta, theta. So you can think about it in the context of ADHD, particularly the inattentive subtype, and autism, and a bunch of other things. We are doing this real-time EEG in our lab. This is one sample. It's actually quite typical in the sense that when we deliver 40 hertz, the brain actually responds in EEG, elevation of 40 hertz. Now, we started with baseline, nothing happened. We put on the cap, and so on, and actually we see a placebo effect. So back to my point, you put something on the head, you actually feel like, people feel like there's something going on and you get a placebo effect. But we went on to look at it in real time, and we have the measures with the EEG plus photobiomodulation. Now we can actually see the difference above placebo, where you can see elevation of gamma, down-regulation of these other oscillations. Then we stop, then everything kind of like cool down and down-regulated 15 minutes, I think, here, and also we measure half an hour, everything goes back up again. It's quite unusual, right? But it shows that there is a longer-term effect. The brain somehow processes the whole thing, and there's a longer-term effect. Now, I always thought that EEG is a byproduct of what's going on in the brain, physiology. These researchers, I think they're from MIT, said brain rhythms are not just byproducts of neural activity, they are critical in organizing it. So that really helped me to process what I'm looking. Then you have gamma in the superficial level. The electrical field from a cell can influence surrounding neurons, and gamma oscillation associated with bottom-up processing rather than top-down. That is also interesting. It put into context a number of other things. So in summary, brain oscillations can be selectively modified by pulsing PBM. Response is quick. We can localize the source of the activity, and brain rhythms have specific characteristics. Let's talk about high frequencies, which seem to be a topic of discussion, not necessarily in neurofeedback field, but very much so in the field of Alzheimer's disease. A lot of work done at MIT, looking at the way it is, say, improving lymphatic flow very recently, or removing beta amyloid deposits and a bunch of things. Now, here's where the location could possibly be relevant. Location is where you position the LED. Now, I'm going to say that targeting the default mode network at 40 hertz will actually start to deactivate it or downregulate it. And when you look at the 40 hertz, it seems to downregulate default mode network, which is really primarily, these are the major hubs like medial prefrontal cortex and posterior cingulate cortex, PCC, and then allow you to move onto the Salience network and then to central executive network, which involves different parts of different regions of the brain. Right here, anterior cingulate cortex, and there's the insular, dorsolateral prefrontal. When the central executive is where you're really controlling stuff. Posterior parietal cortex. And what we're finding in meditation is this is happening when you get into a higher state of meditation bliss, somewhere in between here. So this is a study that's quite well known in the scientific research of meditation. Now, to get into a meditative state, the researchers at Yale, and they're pretty well known actually, and found that the posterior cingulate cortex is actually downregulated. So if you want to get to a higher state, you're looking into downregulating the posterior cingulate cortex. What does the posterior cingulate cortex do? It processes the autobiographical memory. It tells you who you are. You remember who you are, and self-awareness. Well, meditation. You lose yourself and get into a different state. So it seems like this is happening. Now, we did source localization with dipole analysis. We found the same thing. The posterior cingulate cortex is downregulated. The default mode network really is relevant in meditation, but not being stimulated, but actually downregulated. Now we have this study that's ongoing in Toronto. These are preliminary data. We've got 20 subjects, and it's all over the place, right? But the only common factor is we stimulate them with different pulse frequencies. We call it conditions, delta, theta, alpha, gamma, and then we have sham all over the place. But those who are on active generally, we're still analyzing the data, experience through the mystical questionnaire to assess the meditation experience, very significant. But it tells me that it's highly personalized. This is just a sample. The blue actually is delivery of the first condition, which is theta, and this gamma in this person shot up. And this other person, when we deliver gamma, the purple lines, they are slow waves shot up. So not quite sure what we are going to see here, but meditation seems to be a personalized experience. This is actually about Now we develop a prototype of, we call it a NeuroPro, to be able to pulse at different frequencies on the default mode network and see what happens. So this was Sanjay presenting. He was the one investigating, Sanjay Manchanda, a really high-level meditator and teacher. Well, he's talking about studies showing that, in the literature, gamma is pretty high among long-term meditators. Then he tested on pretty renowned meditation teachers and see that they are experiencing this, but they are experiencing it at high frequencies. They are going way beyond 40 hertz, and we're talking about 120 hertz. Sanjay himself today is doing it at 1000 hertz to go into an elevation. He can articulate to you the levels as he goes through the process, the different levels of meditation. This paper is another recent paper. Creative flow looks like it is also involving reduced activity in the default mode network. Like when I talk about the posterior cingulate cortex, is part of it. There's a study done using photobiomodulation and see how it's affecting creative thinking. This is using pulsing at 40 hertz. This came out of the paper, and found that with creative measures, measurement of creativity with the gamma, we found that there's significant improvement, which makes me think that if they were testing on 10 hertz alpha, maybe the results are different. But this was on gamma. Then flow, particularly among the athletes, where you're in the zone, you're performing better. Let's talk about that because it's really in discussion. We're working with some elite athletes and trying to do something about this and see what happens. This was another paper, a look at the flow state, measured with fMRI. And again, they're looking at downregulating of the default mode network. Then, this was very recent, on expert musicians. They do not, summarizing, they do not rely on executive control or the default mode network. They just let go. autopilot. It's kind of like flow, like you stop thinking. I had a discussion with a coach of elite athletes, and he agrees, if we just came out with a slogan, just do it, don't think. Go out there, you've been trained, you spent thousands and thousands of hours, your body is just performing automatically, just perform. And that is kind of getting into a flow state. But of course, you have to do the training. And here's another recent one, looking at actually the electromagnetic field and how it influences electronic gamers. Generally, the diagrams, the pictures are really small, but there are a couple of summary. There is decreased beta activity, high frequencies, in regions of the default mode network, and increased gamma in the regions of the Salience network, which I showed you, default moving the Salience, before it goes to the really thinking, decision-making network called the central executive network. So let's see what can be done about it. There's a collection of some literature, and I'll just highlight that there's, for higher cognition and memory, there seems to be lower alpha and higher fast frequencies in working memory. Higher frequencies are in the top layer of the brain, which kind of go with an earlier study that I showed. And higher processing capacity gives faster response. Okay, your brain is operating as a faster frequency. And this group came up with this, something called neural efficiency in athletes, doing a review. It's kind of saying that, okay, that state is where your brain is functioning efficiently. And I just highlighted a couple. There's alpha-related desynchronization and higher gamma synchronization in the occipital lobe. So that leads us to kind of deliver maybe a protocol for athletes. We have a method now to get into this higher flow state, higher performance, a non-thinking state, a zone down. So it could be a method to deliver 40 Hz and above, down-regulate the default mode network, posterior cingulate cortex, move up to where the meditators are experiencing a higher state, into the salience and maybe a little bit beyond that. We have some elite athletes working with us at the moment, and this is generally my protocol, very simple. Used with the neuro, it can be any device that can deliver the same parameters at 40 Hz before, half an hour to an hour before the event, with the nasal. And then after your event, your training or competition, after that, when you're resting, go to the alpha, try to consolidate, as you rest your default mode network. And that's generally this guy, Ilia Topuria. He won the world featherweight, the UFC title in February, but he was found through his Instagram using this photobiomodulation devices to help him. And later on, I learned that because we knew his coach was somebody, we worked with in Miami. He said, he figured out, use the gamma before, and then later on he was resting with the alpha. Pretty much he figured out, maybe that's why he's at a high level. So that's the result of understanding the parameters of photobiomodulation on the brain. And of course, there's other things like when you're in this sport, you're being bashed on the head and all that stuff. You might have accumulation of trauma in the brain, traumatic brain injury. That if you can resolve traumatic brain injury, I think it helps with your performance. This is an area that we are trying to work with active duty military, some sports people. Alicia Newman, the pole vaulter for Canada, she won the bronze this is her third Olympics. But she always had a brain injury that she walked around with, and that's been resolved, and she got the US got a gold sometime in history, but Canada was the first time, Canada got a medal of any form in pole vault.